Okay. Good afternoon, everyone. Thanks very much for coming to my talk. The topic of my talk today is a cross-species by clustering approach to identifying conserved co-regulated genes. So, because the practical problem motivate us to develop this approach is to identify conserved gene clusters from embryonic development of mammals. So for those of you who are not familiar with embryonic development, I would like to start with a brief introduction of the background of embryonic development. So the embryo development starts from oocyte and undergoes series of developmental stages. So once the sperm enters into oocyte, but before the two nucleuses of the two cells fuse, the embryo is at pronucleus stage. And once the two nucleuses fuse, the embryo is at zygote. So from there, the embryo continues to grow and the cell splits. The embryo will get to 2-cell stage, 4-cell, 8-cell, morula, blastocyst stages in the sequence. So morula stage, the embryo has 16 cells. A blastocyst, the embryo has 32 or more cells. Blastocyst eventually attached to the wall of the uterus, so which completes the pre-implantation embryonic development. So the gene regulation involved in this embryonic development is very complicated and is characterized by the degeneration of maternal RNA stored in the oocytes and also the gradual activation of the embryonic genome. So there are many unanswered questions in this complicated process. The two questions we in our study we're focusing on is, the first one is how genes are co-regulated during this development. So co-regulated basically means regulate together. The genes exhibit similar expression patterns. So the other question is, what are the similarities and differences in gene co-regulation among different species? For example, human and mouse. And particularly, we want to identify conserved, we want to identify co-regulated gene clusters that are conserved across multiple species. So before I get into our approach, I want to briefly introduce the data sets we have in our study. So we have two data sets, one for human and the other one is for mouse. For human, we have data for 6 development stages: oocytes, pronucleus, zygote, 2-cell, 4-cell, 8-cell, and then morula. For mouse, we have data for 6 development stage, including all the stages except including all the stages that human has except a zygote. So across these two data sets, we have 11,018 genes in total shared by them, and so each data set here is basically a matrix. This is an illustration of such a matrix. So each row in this matrix is a gene, and each column in this matrix is a development stage. And the entry in this matrix is the gene expression value at the particular stage. So here the dark color in this, we use the darkness of the cell indicates the magnitude of the expression value. So the more darker the color is, the higher magnitude the expression is. So with this data set, we essentially want to identify the conserved gene co-regulation between human and mouse. So the traditional method that can do this is very simple two-step approach. We first run cluster analysis for each data set individually. Then we are to identify co-regulated gene clusters specific to species, to individual species. Then we run. Then we try to calculate the overlap among the clusters resulted from individual species to identify the conserved gene co-regulation. The major drawback is that, as we all know, cluster analysis is unsupervised. So we may not find considerable overlap among the clusters resulting from individual cluster analysis. So also, potential noises accumulated in the expression quantification process may add to this uncertainty. So our goal in this study is to try to develop a new method for identifying conserved co-regulated genes and by addressing these problems. So our methods includes two components. One is pattern preserving data transformation. The other one is cross-species biclustering method. So let me start with the first component. So pattern especially is a gene expression pattern, which is expression variation across a series of development stages. In our study, each pattern is represented by binary vectors. So 1 indicates high expression level, 0 indicates low or no expression. So if we take the human data as an example in our study, we have this vector with 1 at all sides. All other sides get 0, which indicates a pattern which has high expression level at all sides and low or no expression at the rest of the stages. So whether a gene labeled as G following a pattern labeled as P is determined by their correlation and threshold T. If their correlation larger than T, we see this gene follows this pattern P, otherwise it doesn't. In our study, we use T as 0.75. So because whether or not gene co-regulates determined by the patterns they follow, so we convert the raw matrix or the original matrix, data matrix with raw expression values to a new data matrix with this pattern encoded. So basically, the matrix is a binary matrix, and with each row still represent gene, columns represent patterns that they follow. If the gene follows this pattern, it gets 1. We use green color represents 1. If the gene doesn't follow this pattern, we get 0. We use white color to represent 0. So we use this converted transformed data matrix to do cluster analysis. We avoid to use the raw noise contaminant data to do cluster analysis that reduce the effect of the noise potentially on the co-regulation identification. So with the data at hand, I will start to introduce our cross-species biclustering method to identify conserved co-regulation. So our method is a biclustering method. For those of you who are not familiar with biclustering, biclustering is basically a type of clustering method which simultaneously groups samples and features. So I would like to start from the simple case, which we obtain clusters from single species. And then I will extend our formulation to cross species. So for single species, we essentially give one matrix, data matrix. So actually, our method is based on the sparse rank 1 matrix factorization. So we are given this, for one species, we are given this matrix. So each row represents sample, in our case, is genes. Each column represents features, in our case, it is patterns. So then we look for a factorization of X represented by the out product of U and V. U and V are two vectors. And we have two objectives in this factorization. So we want the product of U and V best approximates this X. And the other objective is we want U and V to be sparse. So we can obtain clusters by looking at non-zero entries in these two vectors. So more specifically, the genes with non-zero entries in U will form correctly gene clusters. The feature or the patterns with non-zero in V will be the pattern the gene in the identical cluster follow. So this is the formulation optimization problem that we formulated. By solving this optimization problem, we can get the U and V. So this optimization problem includes two components. The first one corresponds to approximation error. The second component is corresponding to we want to enforce the sparsity in U and V. So this is a single species sparse rank 1 matrix factorization. Then we extend this to cross species, so we can find the correlation across multiple species. What we do is essentially we add up add together all the individual factorization. So, but except we use a Z trying to link all to unify to all factorizations. And this I is the index of view. We have M total number of views. If we look into this formulation, what we basically do is we decompose U in a single species case into two vectors, Z and UI. Z is shared by across the views. UI is view specific. So here we enforce the sparsity of Z to encourage all UIs has non-zero entries at similar positions. Then we look at the non-zero entries in UI, so to form gene clusters. So genes with non-zero entries in all UIs will form conserved correctly gene clusters, and the patterns they follow can be different from species to species. This is view specific because we obtain those clusters based on VI. VI is view specific. So we validated our method using synthetic data, and we compared with the simple two-step approach that I introduced earlier, and also the several multi-view clustering methods in machine learning literature. So all the methods are measured by NMI. NMI stands for normalized mutual information, and this NMI measures the consistency between the two clusters and the clusters resulted from each method, which ranges from 0 to 1, and the higher the NMI is, the better the consistency is, and the better the method is. So the x-axis are all the methods, and the y-axis are NMI. So our methods labeled as MVBC, and it clearly outperforms all other compared methods. So this is the list of patterns we used in our analysis to do the data transformation. So we include so each row here it represents one pattern. So the dark colors represents 1, the white color represents 0. Totally, we compiled 22 patterns. All these patterns are biologically meaningful, either identified or hypothesized by existing studies. So for example, the first 7 patterns are stage specific. That means all these patterns only has high expression at only one single stage. So these are the patterns we used for mouse in our analysis. In total, we have 18 patterns, and the top 6 patterns are stage specific. So from these data, with our methods, we identified 22 total conserved correlating clusters. Due to the limits of time, I will not be able to discuss all these clusters. I will just show the 2 largest clusters as an example. So the first cluster, we have 1,042 genes, and the patterns that these genes follow are H2 in human, are H12 in human, and M12 in mouse. So this result is interesting because H12 and H12 and M12 are in reverse fashion. So you see, H12 has high expression values in the last 2 stages, but for mouse, M12 has high expression in the first 2 stages. This reveals the difference among human and mouse in their embryonic development in the gene coagulation. So these 2 are the biological functions these genes engaged in. The second cluster has 1,510 genes, and the patterns associated with these genes are H12 and M9. These 2 patterns are similar. They all has high expression level in the last few stages. So this reveals some similarity between human and mouse in their early embryonic development. So these are the biological functions these genes in this cluster has. So lastly, I want to summarize my talk. I just present you our new approach for identifying conserved gene correlations across multiple species. It includes 2 components: pattern preserving data transformation and a new cross-species bi-cluster method. And I have demonstrated our method has superior performance compared to existing methods, and also discuss some of the interesting results from the study on the human and mouse data set. So lastly, I want to acknowledge, I want to thank my advisor, Dr. B, and also my 2 other co-authors from animal science. And I want to acknowledge our funding agency, NSF, and the USDA. And lastly, I want to thank ISCB for the Travel Fellowship. Thank you for your attention.